Thanks very much. Thomas has given us a wonderful introduction to the spirit behind my presentation. I'm going to be talking about one of the work streams that the evidence-based toxicology collaboration has been developing over a number of years now. You'll see that there's quite a shopping list of impressive participants in this activity, and Katja Schön from Thomas's organisation is the leader. And there are others who are the key drivers of this package of work. I've had actually quite a minimal level of technical input to it. I've provided some ideas and some impetus and hopefully a bit of focus to it. But primarily, I'm the spokesperson for a really good group of people who've been undertaking the work. So, out of my line of my presentation, well, Thomas has already introduced DBTC. I'll go through that quite quickly, but also the focus on the drug safety challenge that's led us to undertake this work, and what we've defined as our internal TOX21 project and the data types that we're looking at. And then I'll briefly describe the data types and how we've interrogated them and where we've got to, and put it all together at the end. So Thomas has already highlighted why, first typo there, that's my fault, why he set up EBTC. Really, we need evidence to be able to move away from the system of relying on a paradigm dominated by what I would argue as really good science from before I was born and when I was a young scientist. You know, with the methods we have now and our scientific understanding at the moment, if we were starting again with modern science, we wouldn't be looking to undertake safety testing, and particularly regulatory safety testing, the way we're doing it at the moment. So we need to find a way to move forward from that. We can do it on opinions, and we can do it on our waving and people in glass houses putting up barriers and throwing rocks, or we can actually get together collegiately and say, well, what is the evidence? What is the evidence for what we're doing conventionally with, say, animal-based approaches? What's the evidence for how good they are and how limited they might be? What's the real evidence for these new approaches, in silico approaches, in vitro approaches, so on and so forth? That's the spirit, as I understand it, behind why Thomas set up EBTC. And Thomas, when he set up EBTC, did not want it to be an extension of his brain and his personality. He wanted something that would have independence and governance and oversight. So there's a board of trustees, and I'm one of the members of the board of trustees who work with the collaboration to make sure that the projects we undertake are carefully thought through, that they're aligned and they're undertaken in the right kind of way. So anyway, you've had plenty of time to look at the vision and mission. If you want to know more about EBTC, there's a website link later on. Get in touch with Thomas, get in touch with me, and we'd love to tell you more and potentially work with you, because we're not an exclusive organisation and we look to work with others. It's very much an open opportunity for you to come in and get involved. So, what's the problem? Well, we're all familiar with this, that we know that discovery and development of new drugs is very expensive, it's very slow, and it's colossally inefficient. I used to work in the pharmaceutical industry, and it's just embarrassing how bad we are at taking molecules forward. I never got to work on a molecule that in my time in the company was taken forward as a new drug. It's just a really difficult process. And the problem is it's tough. So there's a lot of barriers. There's the efficacy issue, there's the safety issue, and we have tremendous turnover of compounds. And we're working really hard to try and come up with better ways of doing things. And we've also got the issue that it's really difficult to get drugs approved at the end of the day, but many of the drugs that are approved at the end of the day then go on and cause effects, which in many cases were not anticipated at all from the heroic amounts of work that were done before licensing. So then we get into adverse drug reactions, human ill health, and even more complications. So that's the problem. So we need to be doing something about it, and we need to be applying evidence-based thinking to try and move this forward. So really the challenge that's distilled out here is, well, how can we improve the efficiency, cost and productivity of preclinical drug safety testing? What might we do before the clinical phases, particularly, to give us a much better chance of succeeding in the clinical phases? And also, particularly, one passionate interest I have, what might we do in the preclinical phases to give us a much better chance of having safe drugs when we get to the pharmacovigilance phase post-licensing? Can we be smart? Can we do some things here? And in principle, there's a lot we could do, but it's very difficult to look at look at some of the ideas that people like myself and others have talked about historically and actually evaluate them in an evidence-based way. And that's what this project is all about. And so many people have been developing test methods which potentially, well, they say could be absolutely fantastic. Thomas has just been telling us about brains, for example. Well, what is the evidence that these are actually going to provide us with data that, in particular in a toxicological context, is going to be useful and is going to be more useful than some of the methods that we're trying to displace, particularly the conventional repeat dose animal-based studies, or even more informative than undertaking the early safety studies undertaken in clinical trials on small numbers of individuals. So we've got real challenges on how to work out how to analyse and interpret these new data types. It's great that we've got AI and in silico and so on and so forth, but it's tough, and there isn't an agreed way of doing things. And essentially, I would argue that we need a very efficient workflow that we can apply going forward. And ultimately, we're looking to select a pragmatic number of new safety tests that we can apply for real decision making within companies, within academic labs, and also ultimately in a regulatory setting. So we're not going to do this by complexity, we're going to do this by evidence-based thinking and actually shrink it down to a manageable number of approaches going forward. So that's the ethos behind the EBTC Tox21 project. It's taking account of the fact there are potentially multiple data sources that we would want to be comparing, and how can we extract and interrogate data from these individual data sources in a systematic evidence-based way so we can look and work out how to move forward sensibly. So what we defined for this project after a lot of head scratching is essentially 3 evidence-based work streams. The first is systematically extracting literature data from public domain sources. And essentially this is PubMed type searches, but in principle any publicly available databases are searchable in this way. So if we can get access to the data, we will undertake a systematic literature search strategy. And I'll get on to what systematic searching looks like. It doesn't mean an old person like me going away and looking in a library or doing a Google search. It means systematically thinking about how to do it very well. So you don't bring bias in and you don't overlook stuff and you don't bring in your conclusions of what the outcome should be before you start to undertake the interrogation. So this is really very important. And we've done this for animal-based data and also for human data. And this is human data from published clinical trial data. So we're really interrogating what kind of data might you have before you're licensing a drug, and essentially, particularly, can you match up these animal and human data. We've also got a really creative evidence-based stream. We would like this to extend to a whole range of new human relevant type methods, but for pragmatic purposes we decided we'd go for one big publicly available data source, which is US EPA's ToxCast data set. So we've got a work stream on that. And also a work stream that I really thought was not going to go anywhere at all, and I'm wrong, which was using a pharmacovigilance approach and using the WHO video-based database, which gives us access to a phenomenal amount of post-marketing surveillance information on effects of drugs. And I have to say, this is evidence-based, and we set out to do it that way. If I'd been asked up front, I would have said, well, this, by definition, is going to be largely redundant because an expert can tell you what you need to know about these test compounds. This is going to be really very difficult because it's tough to make sense of a lot of these data, particularly the ToxCast data set. So I'm not quite sure where we'll get from this. And my argument would be, well, this is probably not going to take us very far at all. So that was before we undertook the work. So, for me, this highlights why we need to be doing it in an evidence-based way and why we need to have a systematic project. So there were leaders of the 3 work streams. So Gunvist from the Norwegian Institute of Public Health led evidence stream 1. Sri Bandakavi from Northeastern University, now with Douglas Connect, which is Edelweiss Connect, was the leader of evidence stream 2. And he's fantastic at analysing data. So he did some stuff that I didn't think was going to be feasible. And Johansna Mehta from Caver Health was in charge of the video-based stuff. So, well, I think we already know why systematic reviews are really very important in clinical medicine. They really have been transformative in understanding when you should be using drugs and which drugs you should be using and how you should be using them. So the thinking is, well, how can we apply this methodological interrogation and extraction of data in a toxicological context? So in order to do that, what you need to do is think very carefully about the question that you're formulating, and then you need to get into very careful thinking about the types of data you extract, inclusion, exclusion criteria, and so on and so forth, and then come up with a very careful plan of work. So you need to define very clearly how you're going to undertake the systematic data extraction interrogation before you do any of it. So the aim is that you don't bias the outcome because you do it very objectively. So that's what we set out to do. So a lot of work needs to go into this. And the main study question that we ended up with, well, we were really trying to ask how in vitro tests and animal tests could predict or not liver outcomes that could be observed in humans, either in humans in clinical trials or in humans post-marketing using a pharmacovigilance type approach. So this whole project was formulated, as I understand it, and Thomas can correct me if I've got this wrong, this is really Katya's baby in many respects from a number of years ago, and she was talking to me about this concept. And she very much liked the idea of drug pairs, because we know that there are lots of examples of pharmacologically very similar drugs, one of which is a nasty toxic compound, has been withdrawn or has a strong cautionary label, and another pharmacologically very similar drug, which actually has an excellent safety profile. So can we learn from this? So Katya identified quite a number of drug pairs, and what I'm not describing today is the outcome from a big systematic evaluation of lots and lots of drugs. This is very much a pilot study of a couple of drugs to try and establish, could we develop a workflow that could be applied more generally? And what could we learn about how to do this work so we could do it pragmatically? And we decided that if we started to try and generate data on lots and lots of compounds, we could take forever, and it was much more pragmatic to start on a couple of compounds which we felt we could probably make sense of, a couple of compounds that have been well studied, and then we could take the learning forward and then start to apply it to large numbers of compounds. So that's essentially what we've done. So many of you in the audience here today will know of troglitazone, which was a failed anti-diabetic drug, had a fantastic safety profile preclinically, a fantastic safety profile until it got to very late clinical trials, and then it got licensed and it's crashed out very quickly because it caused really quite a scary number of cases of acute liver failure relatively quickly after its introduction into clinical usage. And it's classified by the US FDA as one of the most drug-induced liver injury concerning drugs. And it's actually probably troglitazone that's been the standout poster child for how the US FDA has completely reformed the way they undertake clinical evaluation of drugs in clinical trials to make sure they don't make that mistake again of taking forward a drug like troglitazone. So troglitazone is a nasty drug, we know that, and we wanted to pull back systematic evidence and particularly compare in vitro and in vivo evidence on troglitazone. Rosiglitazone is a much safer drug. It's not completely safe, but in terms of liver safety, it's got a very good safety profile, people would argue. I would certainly argue that. It's licensed back in 1999. It's classified by US FDA as of low DILI concern. So there have been a relatively small number of cases, but it's been a phenomenally widely used drug. Its usage has been restricted by another toxicity, which is cardiotoxicity. So it's not bland by any means, but it's certainly got a much lower liver toxicity propensity than troglitazone. So both drugs are anti-diabetic. People are gamma agonists, which stimulate insulin function, and the signaling mechanism is down there. So they've got a common mode of action, and they've got a number of chemical similarities up the top here, but also some chemical dissimilarities. So evidence stream one, basically, a lot of systematic stuff. If you get into systematic reviews, you end up with these Prisma diagrams of how it's undertaken. If anybody wants to see all this learning, we're formally writing it up at the moment, and it's going to be published in the near future. And there was a proper protocol that was put up in advance, so we said what we were going to do before we did it. It's a proper system. And the idea was, initially we went through lots of data extraction, and then there was a process of reviewing abstracts, because lots and lots of abstracts came back. Some of them, some of the titles might be completely irrelevant to the toxicity questions we were trying to ask. So there was an abstract selection process, which was tortuous and involved a lot of work from a lot of individuals. And ultimately, we then selected a smaller number of papers, which we decided we needed to go through to full test searching on, and quite a lot of activity. It's actually an incredible amount of data that got extracted from this, and it's all been tabulated and pulled together and analysed. So, really quite an impressive undertaking. And what we were aiming to do is extract data from humans and various animal species as well. So, vast amount of information. So I'm not going to go through all of this in detail, but it will be available in due course. The key thing is we were interested in liver dysfunction, so we extracted and pulled out and summarised and visualised liver parameters, and you can tabulate them or you can come up with a simple colour and visual type representation. So we decided to go for this simple representation. So we've got a comparison between controls, and if you're this side, this means that you're in concern territory, and this side you are not. And we looked at a whole load of different parameters. This is rat serum alanine aminotransferases, one of the markers of evidence of liver injury. So you can see from these studies, for example, this is clear evidence that troglitazone in a variety of repeat dose animal-based studies in rats was actually not making liver function worse. It was actually making liver function, if anything, better, because there was lower levels of ALT release from hepatocytes into livers of rats in these studies. So lots of systematic work was undertaken. So we looked at everything we could extract on animal-based toxicology of troglitazone, and really we couldn't find any evidence of concerning liver signals. So we agree with, I guess, the conclusion that the regulatory people must have gone through at the time that troglitazone was licensed. Based on the available animal data for troglitazone, there's actually no evidence of toxicity. This is ALT data, but all of the other parameters we looked at, we couldn't find any evidence of systematic animal-based toxicity for troglitazone at all. So the animal studies, so this is AST, ALT, so a bit of a wobble here, but nothing to be particularly excited about. So animal-based studies really weren't showing anything at all, and actually something that surprised me is the systematic evaluation of the human clinical trial data as well. The data we were able to access also came up with no signals. So in the interest of time, I'm not showing all of the data, but evidence stream 1 essentially showed that we could not find evidence of adverse liver effects of troglitazone or rosiglitazone in vivo in either the animal studies or the human studies. So this is a compound that was withdrawn because of many cases of fatal liver failure, but this evidence-based evaluation failed to pick out the key signals. So there are clear limitations of the evaluation we were able to undertake. Regulatory preclinical and clinical studies tend not to be published, so we're not actually sure that we got to some of the key information going forward. I suspect that some of the later clinical studies might have been more informative, and that's what I've heard anecdotally and read anecdotally, but we can't say that for sure. We did conclude, and that's why it's in red, that it would be great, and I think really very important going forward, to provide incentives for industry and for regulatory agencies to make these kind of data publicly available in a non-threatening way. And I don't know how that can be managed, but if we can achieve that, that will make future systematic reviews of this type really much more powerful going forward. There was all sorts of stuff around publication quality. If you look at what's published, this is typical of animal-based toxicology. There can be lack of detailed protocols, individual animal data typically are not described in publications, method sections aren't systematic, selective reporting, the abstracts aren't structured in a way. This is a general criticism. If you're trying to undertake systematic reviews, the published scientific literature has not been written in the way that you would like for systematic reviews. So maybe going forward for toxicology, that's something we need to be thinking about, particularly when we're getting into these new evidence streams, because that's what we need to be evaluating going onwards. So the point about structured abstracts. A plea to everybody, myself included, when thinking of publications and peer reviewing publications going forward, structured abstracts are going to be crucial for systematic reviews. So let's see what we can do to improve that going onwards. So moving on quickly, the evidence stream 2, the Toxicology data. Well, this is the largest publicly available database of in vitro test results. It's not a perfect data set, so these are not physiologically relevant assays, but there's an awful lot of them, and they cover lots of different pathways, and many different chemicals have been tested, and you can access the data. So if you're going anywhere, it's a really nice starting point, and it is a systematic data set. So I would argue it's an essential starting point for any compound of interest. If there's data available in Toxicology, start there and then go out from there. So what do we find from it? Well, this was much more interesting from the point of view of a toxicologist who felt that troglitazone should be toxic and rosiglitazone should not be. Oh, I forgot to mention rosiglitazone likewise in evidence stream 1. I don't know, I think I might have edited out the slide. Rosiglitazone did nothing at all in animals or humans in evidence stream 1. So evidence stream 2, well, now we're finding that there are some big differences starting to come through. So we're seeing a lot more signals with troglitazone than with rosiglitazone. And to start off with, it was a binary. Do we see an activity in the assay or do we not see an activity in the assay? In other words, some kind of signal with a potency value associated with it. And troglitazone is lots more active. There's a lot more going on with troglitazone. And another thing is that it's really quite interesting if you look at what kind of biological processes have been activated. Quite a number of the activities were associated with cell cycle, cell morphology, quite a number associated with gene expression, and also quite a number of regulation of transcription factor activity. And I find this quite intriguing as a toxicologist because the real problem we ran into was acute liver failure, and acute liver failure means you've damaged the liver, but also you've impaired the ability of the liver to regenerate. So I suspect that at least some of these are going to be, activities are going to be providing useful toxicological insights. So we've got lots of signals. One of the big issues with troglitazone versus rosiglitazone, which if you're aware of the two drugs, you'll think of first and foremost is troglitazone is a high-dose drug and rosiglitazone is a low-dose drug. So how can we actually take account of exposure? And we had really nice presentations elsewhere in this meeting, and Thomas touched on it as well. We want to get into taking account of dose and ideally getting into quantitative systems toxicology approaches where we consider in vitro exposure versus in vivo exposure and match them up. So we knew that that needed to be done, so we needed some way to take account of in vivo exposure, so the old Paracelsus argument. And the simplest way to do that objectively is to ask, what is the in vivo exposure that's documented for the drugs of interest, and how does this in vivo exposure compare with the potency of in vitro effects? So the simplest way to do it is to get into ratios, comparing the potency in vitro with concentrations in vivo. So a normalised activation score was generated by a bioinformatician who undertook this analysis where he was saying Cmax minus ACT50 divided by Cmax. If I was doing this is not exactly the ratio I would have picked on, but I don't think it matters too much. We have looked at simple ratios between Cmax and AC50, and the outcomes are much the same. So I'm just showing you these normalised activation scores. The key thing is it's a way to start to bin results into unlikely to be concerning, likely to be or potentially concerning, and very likely to be concerning, because if you're starting to get effects at concentrations that are considered likely to be relevant in vivo, then you will be arguing, well, this is likely to be relevant, versus if you've got an effect only at a concentration that's far higher than you could possibly envisage seeing in vivo, that's unlikely to be relevant. Now, I have to say, for this analysis, we were using total plasma Cmax. We were not looking at unbound Cmax. And I think there was a nice presentation yesterday from Liverpool in collaboration with the Satara organisation, where they've started to try and get through this black box of how do we go from total to unbound plasma protein concentration. But the reality is at the moment we're working with total plasma concentrations because it's the closest approximation to what we can work with. It's not ideal going forward. Anyway, we undertook this normalisation. And generated these normalised activation scores for all of our assays. And that was actually quite striking because it really highlighted that there was an awful lot more going on with troglitazone in these in vitro assays than with rosiglitazone, even with the normalisation coming into play. So troglitazone is a much more promiscuous molecule doing a lot more biological stuff in these assays, which potentially could be toxicologically relevant, even taking account of the big difference in doses. So this is some simple heat map visualizations. So you can see this is the target, PPAR gamma, and it's strongly activated by both troglitazone and rosiglitazone, as you would expect. There are quite a number of assay endpoints that were affected in an exposure-adjusted manner by troglitazone alone, and there were a relatively smaller number that were affected by rosiglitazone, but not by troglitazone, and quite a number that were activated by both. So there's some interesting insights into potential mechanisms that have come out from this going forward. So what have we concluded from this? Well, troglitazone triggered 7-fold more off-target biological effects than rosiglitazone, which is really quite striking. So it is indicating that it's a more concerning molecule. and that the drug exposure adjustment has indicated that at least some of these could potentially be relevant in vivo. There are some severe limitations to the Toxicology data. It's not ideal. We would like to look at other data types going forward. And one of the key things that I would home in on as somebody, this is bringing a bit of bias into play, a couple of my key mechanisms for drug-induced liver injury, reactive metabolite formation and inhibition of the bile salt export pump. These are mechanisms not represented in the Toxicology panel. And troglitazone is a potent reactive metabolite former, and one of its metabolites is a very potent BSEP inhibitor. So Toxicology isn't giving us all the mechanistic insight we need, but it's given us some useful indications. But the key thing is it's given us a workflow that we can use to compare with other data types. So moving quickly, the final pharmacovigilance evaluation via Ridgebase. This is WHO International Drug Monitoring Program, started quite a number of years ago with lots and lots of powerful data. So what do we find from this? Well, troglitazone jumps out. This is why troglitazone was pulled shortly after it was licensed. Troglitazone jumps out very clearly as a compound that was causing lots of liver injury, and in particular, lots of cases of acute liver failure. It was only on the market for a few years, and it caused a lot of cases of acute liver failure. And we looked at MEDRA terms associated with troglitazone and rosiglitazone and categorized them going forward, and hepatic failure, hepatocellular damage, clear liver injury stuff, and we use that to help to structure and organise the data going onwards. So, as you'd expect, this links across. The key thing is that when we looked and compared troglitazone with rosiglitazone has been on the market. It's been given to many more patients than troglitazone ever was. And when you adjust for exposure, troglitazone has a far higher number of adverse event associations compared to rosiglitazone. So it's the pharmacovigilance data that's really telling us something informative in real-world terms, whereas the evidence stream one from the clinical trials really wasn't. So I'm reassured by this because as a toxicologist, this is what I would hope to have seen, but we generated this data in an evidence-based, non-biased fashion, which we're quite delighted about. So we like the Ridgebase data, but it's not ideal going forward. But we think it's a really good lead and an indicator of how you can get to real-world scenario data moving onwards. So the overall conclusions, I know we're right at the end of time. Well, in one respect, for evidence stream one, we've confirmed what would have been the pre-existing knowledge, because people would have argued the reason troglitazone was licensed and then withdrawn was because it looked safe in animals and preclinically, and that's what evidence stream one has shown us. And rosiglitazone had a very good liver safety profile, so we've controlled that going forward. And we've also given objective evidence based to the argument that randomized controlled clinical trials simply have insufficient power to detect rare adverse events, and liver failure caused by troglitazone was a rare adverse effect. Evidence stream 2 from the Toxicology data, there's a really interesting new finding. There's 7-fold more off-target effects for troglitazone compared to rosiglitazone, and a lot of potential mechanisms that are above and beyond what a toxicologist like me would have thought could be relevant going forward. So we're very interested in digging somewhat deeper there. And evidence stream 3, again, it's using this pharmacovigilance approach, giving us a structured workflow to enable that to happen going forward. So, really, it told us that animal tests and early clinical trials were not where we need to be, but it's actually given us some evidence-based confidence that these new assessment methods have potential value going forward. So I've said this already. In the interest of time, I won't belabor the point. Next steps are that we're going forward, we're publishing this work, we're interested in following up and going deeper. We'd very much like to move into other in vitro methodologies going forward, and we're really keen to move into other compounds going forward as well. So if anybody wants to get involved in this kind of initiative and they think it could be helpful, please get in touch, and we'd love to get you involved in future iterations of this project. So thank you very much.
Questions from anyone for Gerry? Very apt talk about liver damage the morning after the conference dinner.
Did they investigate the lifestyle conditions? For instance, be responsible for the liver effects? Did they?
There have been some quite detailed retrospective attempts to try and understand what on earth was going on with troglitazone cases. The difficulty is that the failure of troglitazone happened before we knew how to evaluate these cases in the way we would undertake that evaluation nowadays. So a lot of the key clinical samples got lost, and it's very difficult to go back to patients who have acute liver failure because it's got a high fatality rate. So the simple answer to the question is we simply don't know an awful lot of the factors that could have been driving why some susceptible individuals were getting adverse effects and others were not. So we just don't know that, unfortunately. And this kind of analysis probably won't reveal that. What this analysis can focus on is whether a drug like troglitazone would be a favoured drug to take into late clinical trials and even to license in the first place. So that's what we're looking to.
Yes, I was interested by what you found out in that workstream too, around the increased promiscuity of troglitazone relative to rosiglitazone. But I wasn't clear. You mentioned the fact that obviously the clinical dosing is different, but to what extent could the higher clinical dose of troglitazone account for biological effects seen in patients? And to what effect, if you match the exposure artificially, if you like, would troglitazone still be much more promiscuous?
Yes. So if you undertake the exposure adjustment, it's still 7-fold more promiscuous. But quite a number of the activities that were observed in vitro, when you don't take account of troglitazone exposure, you conclude probably aren't going to be relevant because the amount of compound required is so high. It was a high-dose drug. So in modern parlance, that in itself would be quite a big alarm bell. It was a high-dose drug going into large numbers of essentially quite healthy type 2 diabetic patients who really you don't want to put them at risk of drug-induced liver injury. So knowing what we know now, we would be managing troglitazone differently. And knowing what we know now, it would never get licensed by FDA because there would have been ALT signals indicating a potential for problems. So on a cautionary basis, the US FDA certainly would have said no to licensing it at the moment. Yeah, even though we wouldn't have had the acute liver failure cases at that stage.
Thank you, that was a fantastic talk. As part of these streams, would you maybe consider looking at different in silico approaches to identifying the alerts of liver damage or any other sort of mechanistic in vitro models to help elucidate some of those mechanisms around liver injury?
Yes, absolutely. So you're absolutely correct. That's exactly what we need to be doing. So the difficulty in many respects of the evidence-based approach is you need to be quite systematic and careful when you evaluate your data. So if we're bringing in additional approaches like in silico approaches, we need to be very clear about which in silico approach we're going to use and how we're going to generate the data, and have we got a sufficiently robust volume of data that we can undertake a systematic evaluation of it. So for some of these new approaches and some of the new technologies, I think that will be a challenge going forward. The single biggest thing we can do is much better exposure adjustment going onwards. So the quantitative systems toxicology approach, I think, would be really powerful going forward. And we can define a systematic way to do that going onwards. So I'll be pushing hard for us to think of that to help to evaluate data as it's generated. I would love to have access to a large body of data from any of the liver microfluidic devices, if that's available, but I've not seen large amounts of systematic toxicology data from any of those devices as yet. But that would be wonderful.